Hello, everyone. Welcome to Scholar Labs, a one of a kind platform that digitizes science experiments with immersive 3D augmented reality. Our engaging simulations, developed by experienced teachers, enhance learning and are accessible on smartphones, tablets, laptops, and desktops. So, today's simulation is all about plant tissues. In this simulation, you will get a more comprehensive view of the plant tissues and you will be able to understand the vital differences and similarities between the types of tissues. So, before we begin, let's get familiar with some important control buttons. The buttons on the right side of the screen will allow you to view your simulation at different angles. This zoom slider on the bottom left corner of your screen will allow you to zoom in and zoom out. Tissue is a collection of cells that are similar in structure, have a common origin, and usually perform a specific function. So, based on their dividing capacity, plant tissues can be classified into two types, that is, meristematic tissues or permanent tissues. Let's compare their structure and functions one by one. Click on the meristematic tissue to know more about it. Click on this arrow button on the bottom left corner of the screen to expand the menu and view additional features. You can use this button to show or hide the labels. You can also have a 3D 360-degree view of the plant by just clicking on the mouse and dragging it to pan camera angle at different views. We already know that the growth in plants is only limited to certain specific regions. This is because the dividing tissues, that is the meristematic tissues, are present only at these points. These tissues undergo cell division and growth throughout their life. The cells of meristematic tissues are highly active, they have dense cytoplasm, they have thin cell wall and a very large and prominent nucleus, and they lack vacuole. Do you know why they lack vacuoles? This is because these young cells divide actively and require food instantly, so there is no requirement to store food. Also, they do not produce large amount of waste. Depending on the region where they are present, meristematic tissues are classified as apical, lateral, and intercalary meristems. Observe all the meristematic tissues in plants and click on each label to get a close-up view of how it looks. Apical meristems are present at the growing tips of stems and roots. They are responsible for increase in length of the stems and roots. Click here to return to the previous selection screen. Now let's see the location of shoot apical meristem. Next is the lateral meristem, which is present on the lateral sides of the roots. It is responsible for increase in girth of the stem or root. Last one is the intercalary meristem. They are present at the base of the leaf or internodes. They help a plant to grow vertically by adding to its length. Go back to the main screen by clicking on this button to explore the other types of tissues in plants. Now select permanent tissues. Old meristematic tissues differentiate or specialize to take up a specific role and lose their ability to divide and form permanent tissues. Let's have a look at each type of permanent tissue by clicking on the label. So here is a section of stem. You can see here that simple permanent tissues can be classified into three types, that is collenchyma, parenchyma and sclerenchyma. based on their purpose. Let's dive into them individually. Collenchyma are meant to provide mechanical support to the plant structure and have thicker cell walls. Parenchyma, they are living cells with thin cell wall, dense cytoplasm with small nucleus, and a large vacuole. You can see here that they are loosely arranged and have large intercellular spaces. This tissue generally stores food. Yet another type of permanent tissue is sclerenchyma, which makes the plant hard and stiff. The cells of this tissue are dead. They are long and narrow as the walls are thickened due to lignin. Now let's go back to the main screen. Another type of permanent tissues are complex tissues. They are made up of more than one type of cells, unlike simple tissues that are made up of only one type of cells. All these cells coordinate to perform a common function. Xylem and phloem are examples of such tissues. They are the conducting tissues and constitute a vascular bundle. Xylem is responsible for transporting water and minerals from roots to other parts of the plant. They have thick walls stiffened with lignin, and as you can see here, that there is no end wall between the cells. They are composed of tracheids, vessels, xylem fibers, and xylem parenchyma. Phloem is responsible for transporting photosynthetically prepared food from leaves to the other parts of the plant. It is composed of sieve tubes, companion cells, and phloem parenchyma. Sieve tubes have end walls with perforation. Let's go back to the main screen. The other type of tissues present in plants are protective tissues. Epidermis is one such tissue. It covers the entire surface of plant and aids in protection against loss of water, mechanical injury, and entry of germs. So as you can see here that the lower epidermis of the leaves have microscopic openings called stomata, which are surrounded by a pair of guard cells. These guard cells are kidney shaped and contain chloroplast. They are necessary for exchanging gases with the atmosphere and for transpiration. You can have a 3D 360-degree view of the guard cells. Thanks for watching.